Yeah, first I need to say, so my student was invited to give this talk, Jesse Weller, but he can't be here, so I got to talk. So that's good news. But he was supposed to talk just about drug design, but I will give a talk that has two topics because my lab mainly works on protein DNA interactions. They are related, but I will mention both talks, and the second work is by a different student, Rakti Mitra. So drug design first. Jesse developed a method that he called drug hive for drug design based hierarchical variational autoencoder. And the goal of this method is, in contrast to drug discovery, where people look at existing drug libraries and see which of the drugs in an existing drug library binds best to a specific target. With Drug Hive, we wanted to develop a method that designs new chemical compounds that don't exist necessarily in existing drug libraries. And I know in the meantime, there have been some other methods that are related or have a related purpose. But I believe that just being restricted to existing drug libraries is potentially not enough for the future. I will also say using existing drugs has an advantage because these molecules are stable, usually they're not toxic, and they can be synthesized. When you design new chemicals, all of these things are a question. So while I'm showing you this method briefly in this talk, we are obviously aware that some drugs cannot be synthesized and some are toxic, but we are currently testing some drugs that have been derived with this method. So what Drug Hive does, here's the pointer, it looks at a cavity and it basically optimizes the drug that can most efficiently bind in this pocket. By doing this, it will take chemical compound structures and build completely new chemicals, which is shown in the slide after this. But this slide is related to what I said already. So Jesse made this graphic, even though it looks like Wikipedia, but it's not. Here we are comparing the existing traditional method that I call drug discovery. Drug discovery, and Jesse compared the chemical space that can be explored with drug discovery versus the design of new chemicals with the number of water molecules in different entities. Today, existing drug libraries contain about 10 to the power of 8 to potentially 10 to the power 11, maybe soon 10 to the power 15 molecules. So this is basically in this range. And when you compare this to a number of water molecules as shown here on top, then this would represent the mist or a droplet of a spray bottle. These are tiny entities. Whereas when we look at the entire chemical space that is theoretically available by designing chemical compounds that have not been available yet, then this would be Earth's orbit filled with water. So you can imagine, I'm sure the pharma industry works on this, but there are many, many drugs that have not been explored yet. And very likely there are drugs that will change how we treat disease. So this is 10 to the power of 60 of drug-like molecules that can potentially be derived and tested after that. So DrugHive is one of, I said, several methods that design new chemicals. And when we started this, I think there wasn't any method available at that time. Obviously, the AI field races very quickly, and especially in the field of drug discovery and drug design. So what I'm showing you here, this is a variational autoencoder architecture. What I'm showing you here, here's again this our drug target and the cavity where a drug binds. And then we run DrugHive and we optimize the chemical compounds binding to this cavity. I'm showing this in the next slide a little better. We can also determine which area of the chemical compound will be refined. So we can say the blue region here stays constant, we keep it, and we will only refine regions that are either marked in yellow or that are not present. And with that, we can define several tasks that we call linker design. Here you see two chemical compounds that are initially not connected. The method will build a chemically feasible connection between these two compounds. We can do something that we call scaffold hopping, where we change the linker between two chemical moieties of a drug. We can grow the drug, call this fragment growing, or replace patterns in any way that we define or that we want this AI method to find. So basically, the protein structure is the input. This could be an experimental protein structure or an AlphaFold predicted structure. And the chemical and physical interactions that describe the interactions between the protein and the drug will be optimized. And you see this here a little nicer in a video or in 3 little videos where different regions of a chemical compound are refined while this DrugHive method is searching for more efficiently binding chemical compounds. And here is an example where I'm showing you here 2 DrugHive runs. One is based on a protein predicted with AlphaFold 2, and one is based on a crystal structure available in the protein data bank. And you see as a chemical compound being refined or made or optimized, you see on the right for these two structure, the optimization cycles on the x-axis and the negative Wiener score, which is the binding score, on the y-axis. And you see for both DrugHive runs that the negative Wiener score increases, which means binding becomes better optimized. I will say here, and then I'm going to protein DNA interactions. So I envision this as a pipeline. We don't necessarily need the experimental structure. We can use AlphaFold 2 to fold a protein. So we have as input just the amino acid sequence, and then can use DrugHive to predict a drug. As I alluded to in the beginning, a computational method is only as good as it is useful. So currently, we have predicted drugs for a p53 mutant. And in one project, the drug has been synthesized, or multiple variant drugs. And they are now tested for binding, and we seek to have crystal structures with these drugs. In a second project, we also cancer, we predicted drugs. And in this case, we wanted to speed up to functional testing. So after predicting drugs, we went back to drug libraries and ordered the drugs that are most similar to the drugs that we predicted. And those are currently in functional testing, a couple of hundred drugs. With that, I'm going to our other method that I wanted to show you today. We call this method deep PBS, for deep learning-based protein DNA binding specificity predictor. And what they have in common, both methods, they use protein structure. A drug hive predicts a drug or a chemical compound. This method, deep PBS, predicts the protein DNA binding specificity. It uses the structure of a complex. So this is a first step, but the input is requiring to know where the DNA molecule binds, but we will predict the specificity of this protein that is shown here in form of a position weight matrix, or PWM. The height of the letters at the different positions along the double helix indicates how likely or how probable this base pair is, for instance, cytosine and adenine. Then not much specificity at a few positions, and then thymine and guanine. And why is this important? Because when you look at the protein data bank, if you're lucky, for DNA binding protein, you have a complex, but you have always only one sequence, one DNA sequence. In a handful of cases, you might have a complex where a protein binds to multiple different sequences, but that's usually not the case. To get this information, you would need to do high throughput binding assay experiments, such as high throughput CELEX, PBM, or other approaches where you need to refine the protein, bind it in vitro, and then do high throughput sequencing. And after that, some bioinformatics. So it would be a very relatively long path to calculate position weight matrices. We wanted to replace this through a purely computational pipeline where we could predict a complex, for instance, with AlphaFold 3, or take a complex from the PDB that does not have protein DNA binding specificity, and then predict what we would otherwise get only from a high throughput binding assay. This is all written here. Yes, I said all this. It could be an experimental structure or computational structure, and this would replace what you do in high throughut CELEX-seq, or PBM, and other experiments. How does this work? As the idea for this method, we took something that a method that Tzu-Pei Chiu developed. He's here in the audience. He developed another method that I don't have time to talk about, but he basically describes DNA not through the sequence, but through the functional groups, both in the major and in the minor group. And that's actually the chemically more correct representation of DNA. So instead of saying it's a CG base pair, he would describe DNA through 4 functional groups in the major group, here in dark blue, and 3 functional groups in the minor group. This has the advantage that when, for instance, you methylate cytosine, only one functional group in the major group gets replaced by a methyl group. So that means the base pair with a methylated cytosine is very similar actually to CG. But most people who use sequence would just replace the C with, for instance, M, and I would lose this connection between the cytosine and the methylated cytosine, because there would be no information anymore that these two bases are actually very similar and only differ in one functional group. We call this the symmetrized helix. Basically, sequence is removed, And then we use our method DPBS to predict the binding specificity, the identity of each functional group derived from there, the PWM for this given protein. The architecture is shown here. This is a little complicated, but the input here shows we could either use an experimental structure, we could use a predicted structure, AlphaFold or Rosetta fold, or we can also use, and we started doing this MD simulations and use different snapshots of a complex to see if it affects protein DNA binding specificity. Then we represent this is a graph architecture. We represent the protein through a graph where the heavy atoms are the nodes, and we describe them as atom type, charge, and radius to go back to physical and chemical interactions. And the DNA, I explained already, we take the DNA molecule and strip it off the sequence identity, describing it by these 4 positions for functional groups. This is the major groove and 3 in the minor groove. And then our method can determine which readout mode we want to focus on. Usually we use all readout modes. And then our deep learning architecture will learn which are the identities of these functional groups, which will result in binding specificity. In addition, because my lab is interested in mechanism, we also calculate importance scores for each amino acid or each atom in those side chains. And I call it blueberries, but they're not really all blue. They're colored by the chemical element. So blue is nitrogen. And the size of these spheres represents the importance score for achieving the protein DNA binding specificity of the system. We have, in this paper, we have a number of examples, but I want to show one here. This is actually a Rosetta fold NA prediction for a basic helix loop helix transcription factor. And we know that all BHLH proteins bind to E boxes or enhancer boxes of 6 base pairs that are usually CA, TA, TG. And DPBS predicts this PWM as the protein DNA binding specificity of the system. And in this case, we have a JASPA entry, and this is the experimentally measured protein DNA binding specificity. And you see that the central 6 base pairs are very similar. This is the E box, whereas we see some variation in the flanks. And I've done work with Marta Bullock and Aaron Segal years ago, where we pointed out the role of flanking sequences on transcription factor binding specificity. So this could become visible through this method. I mentioned the blueberries. So here's an example, P53. We know P53 has hotspot mutants of amino acids that are in the protein DNA interface. And you see here, for instance, there's an arginine here, an arginine here, and an arginine in the minor groove, which you see here. And you can see that our method identified those important residues as relevant or as essential for protein DNA binding specificity. This is the entire complex, in case you wonder if I know that it has to be a tetramer. So it's just, this is a dimer binding to a DNA half site, and here the tetramer binding to a 20 base pair DNA. This is my last data slide. When we published this paper, the reviewers asked, does this work for synthetic proteins? So we teamed up with Cameron Glasscock from David Baker's lab, and there's already one from the next slide. But what we did is we got designs from David Baker's lab these two proteins, DBP6 and DBP35, and we predicted with DPBS PWMs for these synthetic proteins, which are shown here. And David Baker's lab measured the binding specificity shown in these heat maps. So it's a little counterintuitive, the colors, but white color means this is the preferred, and this is the preferred nucleotide at the position along the double helix. So for this example, in position 8, we have a thymine. We have a thymine up there. Then we have a guanine. We have guanine up there. Cytosine, adenine, cytosine, adenine. So through those measurements, they determined the exact same protein DNA binding specificity that our method had predicted. With that, I'm closing, but I want to mention that we also work on RNA. And one of my students, Weiyu Tang, is here, who has already published a paper that goes beyond this. It's called RNA ProDB, for RNA protein database, that you can use for mapping and also analyzing protein RNA contacts. The paper that we published before that got a cover, so I'm showing it here. In this paper, we mapped, this is a mathematical method, we mapped mathematically the three-dimensional structure of RNA into 2D. And as you know from biochemistry textbooks, when you show RNA secondary structure, you have regions on that map that are far away in the plot, but that are actually interacting with each other. So this new method is called RNAscape for RNA landscape, retains the tertiary interactions and produces these two-dimensional plots. This is the cover for Drug Hive. And with that, I'm showing my lab and my funding sources. So this talk was work by Jesse Weller and Raktim Mitra. Jesse is not here because industry snapped him away. And Raktim joined David Baker's lab a few months ago. And we have a few posters that I'm just mentioning here. And Lexi and Weiyu are presenting posters that are related to the DPBS method that I introduced here. Thank you very much.
So, as I mentioned, next talk is canceled, so we can use time for questions if you have. The next talk is continued as scheduled at 3:40. Questions?
Thank you.
Hi.
Yeah.
I have a somewhat naive question actually. So one of the ways people use models like AlphaFold to get something like a PWM is they, like for instance, say you're interested in 8 mers, then what you do is you just enumerate all possible 8 mers, pass them through AlphaFold, and you can use the DNA side of the LGDT and PAE and other metrics to calculate a position specific, like a motif, a DNA motif.
Yes.
Like if somebody is doing something naive like that, I mean, obviously there are computational limits there, but I was curious, like, would something like that do as well? Has anybody looked at it?
So let me say this. I mean, AlphaFold 3 has not as much data available for making the predictions as AlphaFold 2. So, so you would use AlphaFold 3 and you would do this for a larger number of sequences, but the computational effort would be much larger. You would run an AI method for each sequence. And, you can imagine when you go more than 8 mers, the problem will become very large. And in addition, I envision this, that you use AlphaFold 3 to get an idea about the complex, but that you are not fully relying on this prediction, because there are many fewer protein DNA complexes that are used for making the AlphaFold 3 prediction, which is completely different from AlphaFold 2. So to say that too, we, for instance, recently solved a crystal structure of a fork-head DNA complex, and we did the prediction for this complex before we solved the crystal structure. And AlphaFold 2 told us that the protein folds in a new fold that had not been seen before. And what happened, we solved the crystal structure, and we put this prediction into a supplementary figure 26. Somebody said something about supplementary figures. But when we solved the crystal structure, we actually had this fold, even though it was not seen before. So I'm really a believer in AlphaFold 2, but I'm a little cautious still about AlphaFold 3. It's a long answer, but I hope it addresses the points that you wanted to make.
Professor, I have a question here.
Yes.
Thank you for the talk. When are you using AI that is generating molecules, does it follow what building blocks logic, or the molecule I'm generating sometimes is just not bind or synthesize?
Yeah.
So how is the procedure like in selecting the ligand that would be like the one that I want to?
It designs atom by atom. It's basically an electron density based generation of the new compound. But we designed the method with having chemistry in mind. So our method, at least I can say that, and one pharma company published a blog about this, that our method actually doesn't produce these nonsense molecules that they have seen with our methods. But good point, yeah.
Yeah, thanks for your talk. My question is quite a bit similar. I was just wondering, in the DeepHive method, if you have any optimization on other features that are desirable, like synthesis flexibility, or maybe like penalize potential toxicity or something like that. Do you have something that like explicitly for that, or?
Yeah, that's hopefully our next paper. So, so yes, we extending DrugHive to include those these markers. I want to say at the end, maybe, so I believe in making tools available to the community. And for this reason, so DPBS is a web server that is easy to use, but actually both methods, DrugHive and DPBS, we are now providing them as Docker containers so that people can easily download the method and run it on their computer without jumping through all these hoops of installing, and libraries are being different. So we have, when you go on the GitHubs, we have Docker containers essentially for all our recent methods.
Thank you very much we thank the speaker.
Yeah, thank you.
And